In this lecture, we are going to consider the general principles of the digestive system function and the processes of digestion in the oral cavity and stomach. Digestive system provides the necessary components for the energy production reactions in the body. These reactions are reactions of the biological oxidation. We use carbohydrates and fats as the substrates for biological oxidation, and only in case of their absence, proteins also can be used. Oxidation requires oxygen, and then final products are formed as a result, such as carbon dioxide and water, and of course, energy necessary for life. Oxygen necessary for oxidation is provided by respiration system, where oxygen enters blood, and blood circulation brings this oxygen to cells, while carbohydrates, lipids, and proteins, for all their functions to be performed, are delivered to the circulation system by the digestive system. Finally, circulation system brings these substrates to cells. So the digestive system main function is provision of the organism with nutritional and structural substances. The general digestive function consists of the three main functions. First, for digestion, we need to produce first mechanical processing of food, and also the food which is converted to the chyme. The content of the digestive tract is called chyme. This chyme should be mixed and moved along the tract. Generally, this function can be called, in short, motor function. All mechanical processes are included into motor function, and it is performed by the smooth muscles in the digestive tract. Then, secretion of digestive juices. We need to process not only mechanically but also chemically process the food, and this chemical processing occurs by the enzymes present in the digestive juices and some other substances also. So generally this function is called secretory function of the digestive tract. And finally, when products ready to be absorbed are produced, they should be absorbed to enter blood. And so the last main function of the digestive system is the absorption function. There are many other functions which do not belong to digestion, and these functions are performed by digestive tract. First, we start with, let it be excretory function. Excretion occurs for undigested food substances that remain undigested. Then also there is specific case of excretion performed by digestive system. This is an excretion of bilirubin, substance that is formed after hemoglobin breakdown. This substance is not used as, for example, we use the iron from heme, but heme, after iron extraction, becomes converted to bilirubin, and this substance cannot be reused again, and so it should be excreted. But as it is lipophilic, it cannot be excreted by kidney, which specializes on the water-soluble substances excretion. And so bilirubin has its own way, specific way of excretion. Liver cells take this bilirubin from blood, put it into bile, and with bile it is released into the intestine, where it passes and becomes excreted. Another function, antitoxic function. It's mainly because of the liver function, detoxification of many toxic substances. One more, metabolic function, also predominantly provided by the liver. For example, glycogen formation and then breakdown to release glucose. Then also phospholipid synthesis, cholesterol synthesis, lipoproteins synthesis. Also albumins and other blood proteins formation, all this is performed by the liver. Then protective function. This is function that is provided by many components. Consists of such components like, first, maybe number one is presence of big parts of the immune tissue in the digestive tract. Then also mucosa of the digestive tract contains numerous macrophages, and not only mucosa of digestive tract, but for example liver. Liver macrophages, Kupffer cells, are very powerful macrophages. Then additional moment is the normal intestinal flora, which provides resistance to the pathological flora and performs many important functions for our body. There are more examples given in your text. Vitamins formation. As you know, vitamins are called this way because the body itself cannot produce them, but our intestinal bacteria are able to produce a little amount of certain vitamins for us, such as vitamins of B group and also vitamin K. Centers that regulate our behavior, food behavior. Hunger and satiety center. We already studied this center. They both are located in hypothalamus. Center of hunger is located in the lateral nuclei of the hypothalamus. This center, when it's stimulated, produces excessive eating. Actually, such experiments were done on animals. And for animals, it results in excessive eating, which is called hyperphagia. While destruction of lateral nuclei resulted in absence of hunger sensation, therefore the animal refuses to eat. This is called aphagia. As a result, the animal can even die due to starvation, even in the presence of food, like a plate of food can be present nearby the animal, but animal does not eat and dies of the starvation. Opposite center of satiety is located in the ventromedial nuclei of the hypothalamus. And stimulation of this nuclei in laboratory animals produces absence of sensation of hunger sensation. It results in aphagia, absence of eating, while destruction of center produces absence of satiety sensation. So satiety doesn't come and animal eats practically constantly, hyperphagia. Of course, results of these experiments on animals cannot be directly applied to humans. Human eating disorders are much more complicated, as there are numerous additional factors, including social factors, for eating behavior regulation. And in case of humans, we have such abnormalities of the eating behaviors as the anorexia and bulimia. Anorexia, this term comes from the Greek word orexis, which means appetite. Anorexia, absence of appetite. Bulimia is opposite case when person has very high appetite, or like a bull. The original word comes from appetite, like in a bull. Bulimia. And it can be caused by numerous reasons and sometimes not fully known. Normally, regulation of eating behavior is performed by nervous factors and numerous neurotransmitters and hormones. Let's consider these hormones that are neurotransmitters which are involved in the regulation of hypothalamic centers activity. First, cholecystokinin. This is a hormone that produced in the small intestine, as well as the another substance, peptide YY or peptide Y. They both come from the intestines. They are released in response to the portions of food that come into small intestine, and they stimulate the sensation of satiety while they inhibit the sensation of hunger. Insulin, well-known hormone with its main function to regulate the glucose level, which is secreted in response to glucose increase in blood by pancreas. This hormone, additionally to its peripheral effects, has its central effect on hypothalamic center and also contributes to, as the previously mentioned hormones, contributes to suppression of hunger sensation and corresponding activation of the satiety center. So these hormones decrease hunger. There is one hormone that we know to be increasing hunger sensation. This is ghrelin. This hormone is produced in the stomach, in the empty stomach. When stomach becomes empty, ghrelin secretion increases and it stimulates the hunger while decreases the satiety correspondingly. Also, there is one more hormone that is involved into long-term regulation, leptin, produced by the adipose tissue cells. And leptin secretion depends on the amount of fat deposited in fat cells. This hormone decreases the hunger center activity while increases satiety center activity. But this big difference, it does not operate in response to food. The secretion of this hormone does not depend on the food intake at the moment and presence or absence of food in the system at the moment. It's a long-term regulation. It depends on the amount of fat deposited. And its long-term regulation looks as follows. The higher is fat deposition, the higher is leptin secretion at the moment, and at this level of fat deposition. And it prevents further accumulation of fat by stimulation of the satiety and decreasing hunger center activity. And of course, it also opposes the big loss of fat tissue because its secretion decreases when fat is lost. And so it decreases inhibition of hunger sensation and person will eat more. So in healthy body, leptin provides the long-term regulation of food intake and enables to keep body mass at a stable level, like set point, similarly to temperature somehow, temperature regulation. Let's consider the general principles of the functional regulation of digestive system. There are few types of such regulation. First, it's the nervous regulation by autonomic nervous system. Parasympathetic part of the autonomic system, predominantly vagus nerve, causes stimulation of all digestive function. It increases the motor function, contractions of smooth muscles, and secretory activity of gastrointestinal tract. All juices secretion increases under stimulation from parasympathetic nerves. The only thing that which is inhibited, this is sphincter tone. It means that smooth muscles that compose sphincters become relaxed due to parasympathetic stimulation. Opposite effects are performed by sympathetic part of autonomic nervous system. Motor function and secretory activity decreases by sympathetic part in most cases. And the only thing which is stimulated actually is the higher sphincter tone, opposite to parasympathetic influences. Humoral factors for the regulation of digestive system. There are so many substances that regulate motor and secretory activity of gastrointestinal tract that full list of them will take few slides. But we consider the most important of them. And first to be considered are 4 true gastrointestinal hormones. Hormones mean that they should be secreted into blood. Let's list these 4 hormones. This is gastrin, which is produced in the stomach, as you can see from the name. Cholecystokinin, already mentioned, and secretin, both come from the intestines. And also there is a gastric inhibitory peptide. All these 4 substances are true hormones because they are secreted directly into blood. Then there are a number of pericrine substances, among which mostly mentioned somatostatin and histamine. And there are neurocrines, vasoactive intestinal peptide, gastrin-releasing peptide, and enkephalins. First, let's consider the difference between hormones, pericrine and neurocrine substances. Hormones have the longest way to reach their target cells receptors because hormones are secreted into blood. When they are secreted into capillaries of the digestive tract, they flow within the blood into venous vessels, and then they leave the gastrointestinal tract, pass through the portal circulation through the liver, and only then, by making the full circle, they come to the systemic circulation arteries, where they can be released and reach the target cell's receptors. And these cells are located within the tract itself. Paracrines have much shorter way because paracrine secretion means to secrete to interstitial fluid, per, nearby, and just binds to nearly located cells on their receptors. Neurocrines are similar, but just the difference is that they are secreted by the neurons present in gastrointestinal tract. And release of these substances occur under stimulation by action potentials. But as a result, substances are released and they bind to the receptors of the target cells. The third type of regulation is local or intrinsic regulation. This is regulation by the enteric nervous system of gastrointestinal tract. There are numerous enteric nervous plexuses in the gastrointestinal tract, and these neurons can operate independently of the total general system, and they can change locally the motor activity of the intestine to change the activity of the smooth muscles in the intestinal wall. Then they can change the secretory activity, and they can influence the smooth muscle cells in the walls of the arterioles of the gastrointestinal tract, so they can change the local blood flow. There is the general rule for the regulation of gastrointestinal tract functioning, and this rule is the following. If we start to consider the gastrointestinal tract from its beginning and move down along the gastrointestinal tract, the importance of neural mechanisms decreases gradually, and the importance of the humoral mechanisms and the local mechanisms gradually increases. So you may understand that for oral cavity secretion, for example, for the gastric secretion, even pancreatic and liver secretion, nervous mechanisms are important, but humoral mechanisms become also important as well for the gastric, for secretion of the pancreas. And gradually, when we move to the intestines, the neural mechanisms become less and less important, and for large intestine, actually only local and humoral mechanisms can be important. Let's consider which digestive processes occur along the gastrointestinal tract. The processing of food, which is already becomes converted to chyme, begins in the oral cavity, and it occurs due to mechanical processing by chewing and also by chemical processing due to saliva. Then, next processing occurs in the stomach due to mechanical functions of the stomach and gastric juice secretion. Next, the food goes into intestines, or not food, chyme. Chyme goes to intestines, where it becomes processed due to pancreatic juice, bile, and the own intestinal juice. Therefore, all three juices participate in step-by-step digestion of chyme inside the intestines. And final processing occurs already in the large intestine. For this lecture, we are going to concentrate on the digestion in oral cavity and the stomach. Digestion in the oral cavity. First, let's consider the composition of the secret released into oral cavity, saliva. Of course, it's based on water, but main components include electrolytes, potassium, HCO3 ions, sodium, and chlorine. Notice that underlying potassium and HCO3 are underlined because their concentration is high enough in saliva, while sodium and chlorine concentrations are usually less and mostly much less than their concentration in blood. Mucin, glycoprotein that gives viscosity to saliva, and also mucin performs the protection of the oral cavity. Proteins, such proteins as enzyme alpha amylase, also there is a lingual lipase, and one more protein, also enzyme lysozyme. It provides the protective function, but let's consider all in order. First, how the saliva is formed. Actually, the formation of saliva goes along the same stages as formation of any other juice, because first, acinar cells produce the filtration of plasma, blood plasma, into the lumen. And at first it occurs as a plasma-like isotonic solution. Acinar cells produce this filtration. And at first, concentration of ions is similar to plasma. Isotonic solution then should move through ducts, and ductal cells provide modification of composition. Here, exactly in the salivary glands, the ductal cells produce the following actions. They reabsorb sodium and chlorine. Chlorine simply follows sodium, but of course sodium should be reabsorbed actively. These ions are reabsorbed from the lumen, and at the same time potassium and HCO3- ions are moved into the saliva, a second stage of reaction. As a result, saliva becomes a hypotonic fluid, hypotonic. And the degree of how much is hypotonic saliva depends on the velocity, on rate of formation. When saliva at rest, without food processing, without eating, is formed rather slowly, the flow of fluid through ducts goes slowly, and this reabsorption and secretion processes have enough time to make very low concentration of sodium and chlorine as a result, and therefore the saliva becomes highly hypotonic. When secretion becomes increased with higher and higher rate of formation, and it can be very high during exactly food intake, chewing processes, due to stimulation by parasympathetic nerves, at this moment hypotonicity becomes not as pronounced as before, because fluid flows too fast through ducts, and reabsorption of sodium and chlorine occurs not so much, to such a great extent. And finally, the sodium and chlorine concentration becomes anyway less than blood plasma, but not so much less, maybe 7 times less, as it can happen when formation is not so fast. Functions of saliva. Let's list these functions. First, lubrication of ingested food by mucus, which promotes swallowing. It looks like not of greatest importance function, but imagine what may happen in case of absence of saliva. Swallowing becomes almost impossible when oral cavity is dry, and it can be easily damaged by pieces of food, especially when these pieces are not hard enough. So it's a very important function, lubrication. Then initial digestion of starch and triacylglycerols. This function is not of greatest importance for us. Just minimal amounts of digestion may occur in the oral cavity, and perhaps it's predominantly for taste only. They say if you long enough chew a piece of bread, you can feel the sweet taste. But we don't chew long enough, so much long that finally we feel this sweet taste in the oral cavity. But few molecules can be formed actually, and they can activate the receptors of the stomach, of the tongue, and receptors in other receptors in the oral cavity. And also this is true for lingual lipase. Actually, lipids digestion in the oral cavity is negligible, but perhaps few molecules of fatty acids are formed, and they contribute to better taste of food, and it makes it tasty for us. Supply of calcium ions for teeth enamel. It's very important function, because there is no other source of calcium for teeth. Calcium and phosphates as well. Then protection, extremely important function. Protection of mouth and esophagus by dilution and buffering of ingested food, and also protection by bactericidal properties of saliva. Previously mentioned lysozyme has another name, muramidase. It means that it splits the bacterial wall for many bacteria. So it kills bacteria and helps to prevent development of ulcers, development of different damages for the mucosa in the oral cavity. So due to lysozyme mainly. But also some protective antibodies are present, the so-called secretory immunoglobulin A, present in saliva, and so also it helps to protect the oral cavity and the body as a whole. Now, regulation of saliva secretion. Actually, it's here we remember from the autonomic nervous system that there is no full antagonism of action of the two parts of the autonomic nervous system. Both parasympathetic and sympathetic system actually increase saliva secretion, but of course the degree is very different. Of course, main stimulation of saliva occurs due to sympathetic stimulation. And there are two nerves that can do it. The fibers of the seventh nerve, facial nerve, called the tympani if you remember, they stimulate the submandibular gland and also some other parts. And also glossopharyngeal number 9 nerve stimulates parotid gland, and so secretion of saliva increases greatly. And if you remember, chordae tympani is the nerve which is parasympathetic nerve that dilates vessels of the corresponding salivary glands, so that it produces the basis for greater filtration of blood plasma into the lumen of the salivary glands and contributes to greater production of saliva, chordae tympani. So, as a result of parasympathetic stimulation, high amount of very fluid saliva is formed. High amount because the parasympathetic system is the main stimulating, and fluid saliva is formed because of high rate of its formation. It cannot be so much filled with mucin, and it becomes really very much fluid. And it's necessary when we chew food, especially hard food like dry bread or so. And it should be like this. It dilutes food and helps to swallow the food pieces easily. The sympathetic stimulation. In experiments, it was shown that sympathetic nerve stimulation produces release of little amount of saliva, and this saliva is very thick and viscous, very dense. So many factors which act on salivation. Their action is based on the parasympathetic activation or parasympathetic system inhibition. Due to activation of parasympathetic nerves, the number of factors increase saliva production. And first factor is presence of food in the mouth. It's clear that it should stimulate the reflex of receptors, stimulate the centers of parasympathetic nerve, they're responsible for salivation stimulation, and then food activates the salivation as a result. But also smells, of course, mainly the pleasant food smells. Conditioned reflexes, even when smell is absent, the view of food, which does not stimulate the receptors, but it's a conditioned reflex to produce the high amount of saliva in response to view of food, or even imagination of food, which we don't see at all. We just think about food and salivation may become increased. And all this goes through activation of parasympathetic nerves. And also in case of nausea, stimulation of salivation occurs. And it's a reflex that is directed on the protection of saliva, because the nausea may result in vomiting, and the content of stomach should pass through mucosa, and it's acid, contains acid. But even without this, it produces greater dilution of the content in the stomach, so that it becomes less acidic or less dangerous. So these factors all occur and all act through parasympathetic activation. Then, number of factors inhibit parasympathetic nerves, and as a result, they produce dryness in the mouth, or at least a decrease of secretion of saliva. Sleep. During sleep, there is a little inhibition of the parasympathetic nerves, and during sleep, we have less secretion of saliva. In case of dehydration, it's clear it cannot be stimulated so much. Fear, strong emotions generally, and action of anticholinergic drugs. It's clear because it blocks muscarinic receptors responsible for stimulation of salivation by parasympathetic nerve. So these factors, sleep, dehydration, fear, and sometimes other strong emotions, and anticholinergic drugs, they all decrease salivation. Then we move to the digestion in the stomach. First, functions of the stomach. And surprisingly, perhaps digestion, exactly digestive part of functions of the stomach, perhaps is not the main one. Deposition or storage, it's a very important function which enables us to eat not constantly like some animals have to do, but we can eat 3, 4, or sometimes even 2 times per day, and the supply of content of stomach goes steadily within certain time, and intestines get enough time for processing for a few hours at least, or sometimes longer. So accumulation of food, formation of chyme, this is a part of deposition function, very important. Then mixing of this food to form the chyme, mixing with gastric secretion, mechanical processing which is not finished by the chewing only. It's further making smaller and smaller particles due to mixing. It's also a very important function of the stomach. And finally, part of mechanical function of the stomach is emptying. It's not the final emptying of the chyme from stomach into small intestine. It occurs by little portions periodically, and it provides the regular supply of chyme for further processing and finally absorption. And only in the end we have this secretion of gastric juice, and it provides only initial digestion of food, and mainly this is for proteins, and it's not final digestion, not to amino acids. Amino acids formation is minimal in the stomach. Mostly proteins become converted to smaller peptides, to large or small, or even not small, to large or moderate sized peptides. Absorption in the stomach occurs mostly for water, some fats which are already make very little particles, can be absorbed, but even mostly in babies. And also it's well known that alcohol can be absorbed inside the stomach, but it's not normal substance. Anatomy of the stomach, let's recall a little bit. The stomach has its body, main part, and also the last portion, the so-called pylorus, or it also can be called antrum, and this is intermediate area for transition from stomach digestion to the next part, intestinal digestion. The next part is duodenum. So pyloric digestion is different from main part, from the body of the stomach, as it's a transitional part. And also know that fundus, which itself means bottom, is located not in the lower part of the stomach, but it's the apex, the most upper part of the body of the stomach. Gastric secretion. Gastric juice is produced by glands of the main part of stomach, and it contains pepsin, which is produced by chief cells. Let's recall a little bit of histology. Chief cells produce pepsin, main cells. Then hydrochloric acid is produced by cells that have two names, parietal, and also they can be called oxyntic cells. And mucus, it's an important part of this juice also, and it's produced by mucus or goblet cells. Basically, these are all necessary components for a gastric juice. But as I said from the beginning, that there is a big difference between body of the stomach and a pylorus, pyloric part or antrum. This juice contains only mucus, no enzymes, no hydrochloric acid, no parietal cells. Also, we secrete here hormone gastrin. But no, the gastrin cannot be considered a part of the juice, because gastrin is not secreted into the stomach. It's secreted into the stomach wall vessels, blood vessels. And so it cannot enter immediately. It should pass the whole circle to reach finally the cells it influences. Now let's see this main type on the scheme. First to be mentioned are chief cells, and you remember they produce pepsinogen. Pepsin, main enzyme of the stomach cells, is produced in its inactive form, pepsinogen. Actually, there are a number of slightly different pepsins, and we can say in multiple pepsins. But basically, they have just a little difference in the optimal pH. Slightly, the range of pH values is different for subtypes of pepsins. We can say simply pepsin as one single enzyme that operates in acidic medium and requires the acid for activation, because conversion of pepsinogen into active pepsin occurs due to hydrochloric acid, and hydrochloric acid is released by parietal or oxyntic cells. But note that these same cells also produce one factor, not the digestive factor, but the so-called intrinsic factor. We'll discuss intrinsic factor on the next slide, but generally, I hope you remember that this factor is necessary for absorption of vitamin B12. And then mucous cells. They are present everywhere in the stomach, and only mucous cells are located for formation of juice in the antrum or in the pylorus. Mucus is produced by these cells or goblet cells. And as for pylorus, also there are endocrine cells, G cells, gastrin producing. G stands for gastrin. And G cells produce gastrin to circulation. You see, it's not directed to the stomach juice. No, it's not a component of juice at all. It's secreted into blood. And of course, it becomes moved by blood into venous vessels and into venous side of capillary, and finally it leaves the stomach, and with circulation it passes through the whole circle. It means that it passes through portal vein, then go to the pulmonary circulation, then finally enters the systemic circulation, and through gastric arteries it reaches the parietal cells of the stomach body. And gastrin provides its main effect by stimulation of parietal cells, and it increases secretion of hydrochloric acid. So when some parts of food, some content in the substances in the chyme stimulate G cells, they secrete gastrin in response, and gastrin stimulates secretion of hydrochloric acid in the stomach body. The factors are to be listed next. So, hydrochloric acid is stimulated, its secretion is stimulated by gastrin. So it's produced in the stomach and influences mainly the stomach, just different areas. Gastrin itself is produced in the pylorus only, and parietal cells are located in the body of the stomach only. Secretion of the intrinsic factor. Let's come back to another factor produced by the parietal cells. As already it was mentioned, it's essential for absorption of vitamin B12. Vitamin B12 first is protected from destruction by the hydrochloric acid in the stomach. Its first intrinsic factor binds to vitamin, and as a result it's not destroyed in the stomach. But then the complex of factor and vitamin B12 goes further into intestines. And finally, this complex, vitamin B12 intrinsic factor, binds to the specific receptors in the distal ileum, in the end of the whole small intestine. And as a complex, these substances become absorbed. So vitamin B12 is considered to be the external factor that comes with food, but intrinsic factor provides finally its absorption. If there is no vitamin B12, of course nothing to be absorbed, but if the vitamin is present, but there is no intrinsic factor, absorption does not occur. So in cases of, for example, of atrophic gastritis, where the cells are lost that produce both hydrochloric acid and intrinsic factor, the absorption can be not occurring, and as a result there will be deficiency of this vitamin. Also in case of total removal of the stomach due to some medical indications. Also, there is a problem that vitamin B12 can be deficient. We have great storage of this vitamin in the liver, and it can be enough for even few years sometimes, but finally the storage can be depleted. And if you remember, B12 is necessary to prevent formation of anemia, megaloblastic anemia, that can develop in case of long-term deficiency of this vitamin. Mechanism of gastric secretion of hydrochloric acid. This is an important question, and this mechanism is necessary to understand. To produce H plus ions, and as well, actually, finally, chlorine ion follows it to produce hydrochloric acid. First, we need to secrete H plus ions into the lumen of the stomach. Let's consider the parietal or oxyntic cell in the stomach wall. They are scattered around the regular epithelial cells. One side, the left side now, is a capillary, and the other side is a lumen. And H plus ions really comes from water molecule. Look, reaction of combination of two substances is used for secretion of hydrochloric acid. This is usual reaction of carbon dioxide binding H2O, water. As you remember, this reaction itself goes very slowly, but we have the widespread enzyme that produces this reaction by increasing this reaction rate by few thousand times, or maybe 1,000, as is known from biochemistry. And so CO2 sometimes, own CO2 production by cell sometimes becomes not enough, and therefore additional CO2 is taken from blood. Depending on rate of secretion of hydrochloric acid, CO2 requirement can be different for parietal oxyntic cells. So, first, carbonic anhydrase produces the carbonic acid, which becomes split into H plus and HTO3 minus ions. And exactly these H plus ions we need to form the hydrochloric acid. First, HTO3 minus ions are removed from the cell due to exchanger. Exchanger, as you remember, does not use ATP. Chlorine HTO3 minus exchanger provides entry of chlorine into the cells, while H2O3 goes into blood. And for transportation of hydrogen into lumen of stomach, the cells use the pump. And now this is the using ATP transport. So it's a pump, active transport. It's very unusual pump, which haven't been mentioned before. H plus, K plus pump. Hydrogen, potassium pump. And operates the following way. H plus is transported into the lumen, while potassium, against concentration difference, is transported into the cell. Hydrogen potassium pump. Sometimes it's called simply proton pump because H plus ion is actually proton. So H plus is transported. Chlorine, which due to exchanger activity appears in the cell, simply follows H plus with its minus charge. And as a result, both ions appear to be in the lumen of stomach, and so it makes the hydrochloric acid. And HTO3 minus excess in venous blood that flows from the stomach produces the so-called alkaline tide in venous blood that passes through stomach veins. It has much more HTO3 than average blood. This mechanism is responsible for formation of main component of the gastric juice, one of the main components of gastric juice. Let's see how these cells look not schematically but closer to reality. You see, their lumen is hidden partially and goes to the surface of the stomach mucosa. To the right side it's a lumen again. And H plus ions as well as chlorine ions move gradually into the lumen and finally appear to be on the surface of the stomach mucosa. And stomach mucosa is covered by mucus, so that hydrochloric acid normally does not contact the epithelial cells and does not damage them. Also these cells produce intrinsic factor necessary for vitamin B12 absorption. Stimulation of gastric hydrochloric acid secretion, or simply gastric H+ secretion. Which factors are responsible for this? First, these cells are innervated by vagus fibers, parasympathetic stimulation, vagus, by acetylcholine release and binding to muscarinic receptors on the parietal cell membrane. Then gastrin. Gastrin release from G cells of the pyloric part of the stomach. Specific stimulating factors for gastrin release are small peptides and amino acids. So when they appear, it means that the stomach content, the chyme, contains proteins, and proteins require much more acid for their digestion. So when first portions of small peptides appear in the pyloric part of the stomach, then gastrin is released in high amount than usually, and it stimulates cells producing hydrochloric acid stronger. Then distension of stomach, stretching of the stomach, also stimulates gastrin secretion from G cells. And vagal stimulation via gastrin releasing peptide. So vagus also stimulates G cells, but in contrast to stimulation of the cells that produce hydrochloric acid, of parietal cells, by vagus, there is no acetylcholine secretion, but instead of acetylcholine, which is usually secreted, gastrin-releasing peptide is released, and it stimulates G cells to produce more gastrin. And also inhibiting factor. Inhibiting factor is easy to predict. It's a negative feedback factor. Gastrin stimulates H+ secretion, but excess of H+ ions logically should inhibit further secretion of gastrin. And really, they inhibit gastrin secretion. Histamine. Histamine is released from mast cells in the mucosa, and it also stimulates the secretion of hydrochloric acid by activation of specific histamine H2 receptors, which are located on the parietal cell membrane. Let's consider all the factors that affect hydrochloric acid, or simply H+ ions, gastric secretion. Parietal cell final mechanism of H+ ions secretion is pump, hydrogen potassium pump. This pump is stimulated by number of factors that stimulate secretion. First, acetylcholine. It binds to muscarinic M3 cholinergic receptors. When acetylcholine binds to these receptors, these are 7-TMS receptors as usual, and they operate through phospholipase C pathway. So IP3 is formed, inositol triphosphate, which increases calcium concentration, and this results in high activity of the pump. Next factor is histamine. Histamine binds to H2 histamine receptors on the membrane of the parietal cells. And this binding activates the pathway through cyclic AMP, also 7-TMS receptors that activate adenylate cyclase. Cyclic AMP formed finally activates through a number of stages the activity of the pump, increases activity. Gastrin. Gastrin also has receptors located on the membrane of the parietal cells, and when gastrin molecules bind to these receptors, as a result, it increases somehow concentration of calcium, which also stimulates the pump activity. So, all these factors are stimulated. An additional mechanism is potentiation. It means that when each factor acts alone, it produces certain effect. But when the two factors or more act together, they provide effect higher than summation of the two alone. For example, gastrin increases the sensitivity of histamine receptors, and action of histamine becomes higher. Acetylcholine and histamine mutually stimulate and increase effect of each other. and presence of acetylcholine influence and histamine increases gastrin activity. Generally, each factor added produces high and high effect. Therefore, even if we inhibit one of factors, it decreases the degree of potentiation and total effect of stimulation can become less. When it can be necessary? When there is certain damage of the mucosa in the stomach. For example, if there is a gastritis or ulcer, it's necessary to prevent further damage, which can be produced by any presence even of acid in the stomach. And in these cases, we need to inhibit, to decrease hydrochloric acid formation in the stomach. So usually it's the main task of the pharmacology to suppress the secretion of the acid by stomach. And how we can do it? Factor that decreases. We can block any of these receptors, except for gastrin, not identified still. For block of action of acetylcholine, we have blockers of muscarinic acetylcholine receptors, which the main example is atropine. So, atropine, when it blocks the receptors, prevents the effect of acetylcholine. And potentiation effect of acetylcholine also is decreased. But generally, pharmacology does not use this way for suppression of acid secretion because muscarinic receptors are widespread in the body and there will be numerous additional effects which are side effects mainly. So it's not a widespread type of decrease of hydrochloric acid secretion. More often, we use the blockers of H2 histamine receptors. Such a drug as cimetidine is widely used for this purpose. And when histamine H2 receptors are blocked, effect of histamine is decreased, effect is lost, and its potentiation effect is also lost, so that less activity is produced by gastrin and acetylcholine. And for gastrin, there is no blocker developed, but we know how to suppress the secretion of hydrochloric acid regardless of initial mechanism of stimulation, because we can block the final mechanism, which is pump activity. And most often what is used for suppression of gastric secretion is blocker of pump, sodium or for hydrogen potassium pump, or simply proton pump. The main drug developed for this is called omeprazole. It can have different commercial names, but basically it's omeprazole, which blocks the pump, and finally it doesn't matter which factor stimulates the parietal cells. The final mechanism of production of acid is blocked. So omeprazole is effective drug to suppress the secretion of hydrochloric acid in the stomach. Functions of hydrochloric acid. Fortunately, for healthy person, we don't need any suppression of secretion of this acid. It's important for digestive purposes, and not only. Let's list the functions of hydrochloric acid. First, denaturation of proteins. Proteins have complex structure, not only primary structure, which is a chain of amino acids, but it has the spiral shape, and also this spiral shape has the secondary, tertiary structure, or even above this. And all these additional structures, shapes of protein is lost, so they become simply converted to a chain of amino acid by action of hydrochloric acid. And this preliminary action greatly helps for pepsin to start cutting the protein chains into pieces. First stage of protein digestion simply. Then pepsinogen should be converted to active pepsin, and this requires hydrochloric acid, H+ ions. But then, even if pepsin is already formed, it needs, for active functioning, it needs the acidic medium. For pepsin, pH optimum is between 1.8 up to 3.5. It includes all the variety of pepsins, or simply we can say one pepsin, and its optimal functioning requires this range of pH values, which can be provided by acid only. Then bactericidal action. This is important protective function of the hydrochloric acid. Your food not always is free of different bacteria, And also you remember that we have swallowing, regular swallowing of mucus from respiratory airways. And also this mucus can contain bacteria that can enter with the inhaled air. So bactericidal action is very important. Most of bacteria are killed due to hydrochloric acid action. And number of functions, the stimulation type. Stimulation of intestinal motor activity. It contributes to greater degree of smooth muscle contractions. Then secretory activity also increases. And not only of own intestinal juice, but also hydrochloric acid. Greater amount of this acid stimulates through specific humoral factors, higher secretion of pancreatic juice and higher bile secretion. So there is a number of functions performed normally by hydrochloric acid. Acid secretion can be very different depending on situation. When stomach is empty, it's minimal. But when food comes into the stomach, it increases secretion of hydrochloric acid. And if this food contains not only carbohydrates and fats, but also contains a lot of proteins, the more proteins are present, the higher hydrochloric acid secretion is. Protection of gastric mucosa from damage. Why normally the stomach mucosa doesn't have any damages due to hydrochloric acid? First, because it's covered by mucus, and because this mucus contains a lot of HCO3- ions. Here is a schematic presentation of the stomach wall cells layer covered by the mucus layer. And within this mucus there are a number of HCO3- ions. They are main protective factors. So you see, normally gastric mucosa is not directly exposed to highly acidic gastric juice. So mucus and HCO3- are main factors. But nearby in the table you can see the other factors, such as prostaglandins, the presence of mucosal blood flow, and the number of growth factors secreted by cells. Specifically, you should pay attention to prostaglandins. And prostaglandins, there are a number of some types of prostaglandins that are responsible for protection. And also you can understand why among damaging factors we have this abbreviation NSAID, non-steroid anti-inflammatory drugs. Of course, among damaging factors you can see stress, smoking, alcohol, presence of Helicobacter pylori, and of course the H+ ions themselves can be damaging with pepsin. But NSAID, why they are responsible also for damage? Because you remember they operate through the inhibition of cyclooxygenase, which blocks formation of prostaglandins. And among these prostaglandins, not only pro-inflammatory ones are present, but also the favorable prostaglandins that provide protection of gastric mucosa. And when these substances are used, especially for a long enough time, then they block prostaglandins formation and decrease protection of the gastric mucosa, so that it's often happening side effect often occurring. The damage of the mucosa and even ulcers can appear due to long-term usage of non-steroid anti-inflammatory drugs. Final question is about regulation of gastric secretion, and it goes in phases. This picture looks complex from the first sight, but we can divide 3 phases of gastric secretion into 3 simple ones: before food enters the stomach, when food is present in the stomach, and the third phase, when food goes out of the stomach. So it's very easy. Before food enters the stomach, there is also already increased gastric secretion, not greatly, but increased. This phase is called cephalic, because it comes from the head, from oral cavity mainly. And this cephalic phase is subdivided into 2 parts. First part occurs before food acts on receptors of the oral cavity, so before anything is present in the oral cavity. And all this phase goes through vagus nerves. So first goes as a conditioned reflex. View food, smell of food, or even imagination of food already increases not only salivation, which we can detect easily. Пожалуйста, скажи, что не могу. And this is a conditioned reflex which occurs due to early learned response, because the newborn babies do not have this effect of stimulation, but very early it becomes studied, and then it becomes fixed as the conditioned reflex. So cephalic phase begins from the first conditioned reflex, but when first pieces of food already start to act on receptors in the oral cavity, as a result, it stimulates the vagus fibers as well, and additional secretion of gastric juice, preliminary secretion, occurs. It's not great secretion, but already stomach prepares itself for taking food and further its digestion. Second phase is a gastric phase. During this phase, maximal secretion occurs because exactly at this time we need to have more gastric juice. And here mechanisms include local secretory reflex due to stimulation of local receptors present in the stomach mucosa. Also due to vagus reflexes, not local, so it means sending impulses up to the brain, to vagal nuclei, then back from vagus. Efferent nerves bring stimulation of secretory cells. And the third mechanism is gastrin and histamine stimulation. Gastrin is secreted when first pieces of food come to the pylorus, where mechanical stimulation, stretching, additionally present of protein degradation products like peptides or even some amino acids. And these factors activate the gastrin secretion, and gastrin secretion increases the secretion of this acid in the body of the stomach. And histamine cells are present throughout the mucosa. Also histamine is released, and it contributes to greater secretion of acid. Some endocrine cells in the mucosa present that secrete histamine as well. So during this phase, maximal secretion occurs. And when the third phase begins, when first portions of the food start to leave the stomach due to stomach emptying, and this mechanism gradually decreases activity of gastric secretion, and when last portions leave the stomach, this secretion becomes less. So three phases: cephalic, which consists of two parts, then gastric phase, and intestinal phase, which predominantly decreases the secretion. Nervous and hormonal mechanisms are involved in the third intestinal phase. So thank you for attention. This is the end of first part of digestion.